Welcome back. Today we're going to be talking about the production of gametes, and that is spermatogenesis in the male and oogenesis in the female. We're going to be starting with the male side because it's just a little bit easier. Remember that spermatogenesis happens in the seminiferous tubule. And this picture shows a cross-section of a seminiferous tubule right here. So this is the outside portion, and you can see the inside portion. The lumen is right there. And the spermatogonia are going to start on the wall of the seminiferous tubule, and as we go through spermatogenesis, the developing spermatocytes are going to be moving from the outside towards the inside. You can actually see the immature sperm right here with their flagella or tails hanging into the lumen of the seminiferous tubule. And we have two kinds of cells that are going to help us. The first are the Sertoli cells, which you can kind of see right here. Those are cells that are within the seminiferous tubules, and their job is to support and nourish those developing sperm. And they also produce a hormone called inhibin, which is important in regulation of hormones. The second type of cells are the interstitial cells, and they're the ones that are going to produce testosterone. And you can find those sort of between the seminiferous tubules. The testosterone that they produce is going to help stimulate more sperm production. So before we get into the nitty-gritty of how this all happens, let's talk a little bit about the hormone regulation of spermatogenesis. And for that, we have to go way back to the endocrine system. Remember the hypothalamus and how important it was in maintaining homeostasis of many different systems in the body? Well, sexual function is one of the systems that the hypothalamus is important in. It produces a hormone called gonadotropin-releasing hormone, which acts on the anterior pituitary, and the anterior pituitary produces 6 hormones, but in response to gonadotropin-releasing hormone, it's going to produce luteinizing hormone and follicle-stimulating hormone. Now, follicle-stimulating hormone is going to act on those Sertoli cells. Remember those cells that are supposed to be helping to sort of nourish and help and take care of the sperm. So follicle-stimulating hormone is going to act on those cells and cause them to help push the cells towards spermatogenesis. Luteinizing hormone is going to act on a different kind of cell, the interstitial cells. And it's going to tell those interstitial cells to produce testosterone, which also helps to stimulate production of sperm. Now, this is regulated by a negative feedback regulation. So, for example, as the level of testosterone starts to rise, it is going to feed back to the hypothalamus and the anterior pituitary, and it's going to shut down gonadotropin-releasing hormone and luteinizing hormone production. As those two hormones fall, we're going to have less stimulation of the interstitial cells, and so testosterone will then fall. Likewise, as you start to get more and more follicle-stimulating hormone produced, the Sertoli cells, which are being stimulated by the follicle-stimulating hormone, are going to produce something called inhibin. That inhibin is going to feed back and shut down gonadotropin-releasing hormone and follicle-stimulating hormone. So again, there will be less follicle-stimulating hormone produced, which causes lower triggering of the Sertoli cells that are going to push spermatogenesis. As both of those things start to fall, we can get rid of this negative feedback, produce our gonadotropin-releasing hormone, and produce our follicle-stimulating hormone. So unlike the female system, in this way we are able to maintain a pretty consistent and stable level of sperm production. Every time the level gets too high and the entire system gets too triggered, we can turn it down. If it starts to fall too low and we're not making enough sperm, we can juice it up again. And so that's what is important about this negative feedback. So men, once they hit puberty, begin to produce luteinizing hormone and follicle-stimulating hormone, begin to produce the testosterone that causes those secondary sex characteristics in males, more hair, more muscle development, lower voice, that will be maintained throughout life. So men do not go through something like menopause, which I guess would be menopause. They don't go through that. They continue to make testosterone throughout their life and only have a gradual slowing down very late in life. So men who are 80 years old can still father babies. Okay, so let's talk about spermatogenesis. So again, spermatogenesis happens in the seminiferous tubules, and it takes about 74 days to go from the very beginning to the end. And the first thing that's going to happen is mitosis. The spermatogonia, which are on the walls of the seminiferous tubule, are going to divide, and it's going to form two daughter cells. One of them is going to go on to become sperm, and the other one is actually going to replace the spermatogonium. So it's going to make another cell that can then divide again and keep on populating. So this way we can always produce new sperm. So that primary spermatocyte is a diploid cell, and it's going to go through meiosis 1, and we're going to go from a diploid cell to a haploid cell in our secondary spermatocyte. You can see our Sertoli cell right here is right near all of these cells, helping to support and nourish them. That secondary spermatocyte is going to go through meiosis 2, and each one of these daughter cells will produce two daughter cells. So you're going to end up with 4 early spermatids. Now, those spermatids don't really look much like sperm. Although they've already gone through meiosis and gone from a diploid to a haploid cell, they still have to go through a lot of morphological changes that can swim and get to the egg and then penetrate it. And this last part here is actually called spermiogenesis. And that isn't any mitosis or meiosis. It's really just changing of the structure of the sperm. That takes about 24 days for that to happen. The entire spermatogenesis took 74 days, and this 24 days is the last 24 days of it. So spermatogenesis includes mitosis, meiosis 1, and meiosis 2. Spermiogenesis only is the last part of that. Once the sperm are fully formed and have the right structure, they will be stored in the epididymis until ejaculation. Okay, so we've already talked about spermogenesis regulation, and it's a little bit different in the females. Although it is similar hormones, it happens on a cyclical basis, meaning every month we're going to have the same cycle happening. And that cycle was talked about in the ovarian and uterine cycle video. So remember, we still have the hypothalamus. It's still producing gonadotropin releasing hormone, which acts on the anterior pituitary, which causes the production of luteinizing and follicle stimulating hormone. In this case, follicle stimulating hormone is going to act on the follicle of the ovary and cause it to mature one oocyte, and luteinizing hormone is going to act on the ovary to cause ovulation. After ovulation, the corpus luteum is going to produce progesterone and estrogen, our old friends, that their job is to maintain and thicken the uterine lining. In addition, progesterone and estrogen are going to feed back and shut down the production of not only gonadotropin releasing hormone, but also luteinizing hormone and follicle stimulating hormone. And we do this because once that egg has been ovulated, it might be fertilized, and we don't want to immediately start maturing another follicle until we find out if the egg that we just made is going to be fertilized and become a developing embryo. If you don't remember how all of this works, please go back and rewatch the ovarian and uterine cycle video for a more in-depth explanation. So, oogenesis. This is the picture that is in your textbook, and while it is correct, I feel like it's missing some key details. So I'm going to use something that I found on the internet, and just has a little bit more specifics to it. So oogenesis starts in a developing embryo. And all of the oocytes that a woman is going to produce have already been formed in utero. So all of these primary oocytes have already been formed before that woman is even born. And those oocytes are arrested in prophase 1. So they just stop there. And if you remember, prophase 1 is the phase where we have the chromosomes are synapsed and crossing over is happening. And then that's it. We don't have any spindle fibers reaching out. We don't have anything lining up anywhere. That's the primary oocyte. Now, we're going to wait somewhere between 12 and 13 years during childhood for anything to happen. Once they go through puberty, a woman is going to start producing follicle-stimulating hormone. And follicle-stimulating hormone is going to act on the oocyte and the follicle and cause maturation of one follicle. That oocyte is going to finish meiosis 1. So remember, it was stuck in prophase, so it's going to go to metaphase, anaphase, and telophase. And during cytokinesis, we're going to have an unequal distribution of all the stuff that's in the egg. So the majority of the cytoplasm, the organelles, the mitochondria, all the stuff is going to go into one of the daughter cells. The other daughter cell is almost like a cellular trash can. We're basically just going to put those extra homologous chromosomes, the ones we don't want, into the other daughter cell. And that's actually called a first polar body. And it ends up just dying. We're not even going to use it anymore. Now that secondary oocyte is going to start meiosis 2. And this is where we're going to have our big surge of luteinizing hormone. And the secondary oocyte is going to be ovulated from the ovary. Once it's been ovulated, it's going to enter the uterine tube and maybe fertilization will happen. If fertilization happens, we're going to finish meiosis 2. It got stuck in metaphase 2 and it won't continue unless fertilization happens. Once fertilization happens, we're going to have another unequal distribution of the cytoplasm during cytokinesis. So we're going to make a second polar body, which is just going to be those extra chromosomes, those extra sister chromatids that separated in anaphase 2, and that polar body is going to die. And this zygote, remember a zygote is a fertilized egg, is going to go into the uterus. So remember the corpus luteum, which is sort of the part of the follicle that survives after ovulation, is going to produce our progesterone and estrogen, which are going to act on the uterus to thicken and maintain the lining. And so if that fertilized egg gets there and the lining is nice and thick, it's going to be happy and want to implant in the uterus. However, if fertilization doesn't happen, then this secondary oocyte will not complete meiosis 2, and none of this will happen. And very quickly, if there's no implantation and no production of HCG, human choriogonadotropic hormone, which is what we look for in a pregnancy test, then the corpus luteum will regress and stop producing our progesterone and estrogen, and the lining will be shed along with the unfertilized egg. That's it for today. See you in class.